So namaskar. So thank you very much again for this opportunity to present a few of my observations during this pandemic and the scientific, from a scientific angle. So let me. I'm going to mainly speak about the process of developing the vaccine based on the understanding of immunology. मेरा background है, मेरा काफी immunology में background है, तो immunology के angle से मैं इस pathogenesis को देखता हूँ. So the topics I will cover are obviously the introduction, the basic immunology of the virus, the components of the virus. और आपने कल COVID के बारे में सुना है, तो I won't spend too much time on the components. The immune response, बहुत interesting immune response है इस virus का. Very interesting things are happening with this virus. I'll talk a little bit about the process of how a vaccine is made and what the rationale for making this vaccine is, and then I'll end with what is the herd immunity. So I will cover these topics. So the scientific process, and I'm told that a lot of you are students. The scientific process in anything is the ability to observe everything very minutely and very clearly. So in the turn of the century, when Edward Jenner was discovering the smallpox vaccine, a very interesting observation was made. Milk maids and milk men, although they were all being staying in the same village, were not infected with smallpox. Even though the rest of the whole villages and towns and cities and many thousands and millions of people were dying of smallpox, milkmaids were not. Nothing was happening to the milkmaid, and they had a beautiful fair complexion, and nothing used to happen to them. So when Edward Jenner made this very keen observation, so why are they not getting infected? He discovered that the virus from the cow was being transmitted to these milkmaids, and that's why they were getting protected. So that's when the theory of vaccination, the modern vaccination, started, of developing vaccines to protect us from infectious diseases. It came from a very simple, very keen observation of what is happening in the surroundings. So similarly, we have to observe the immune system, and we have to observe what is happening with the virus in the world today very scientifically, to get us clues as to why or what we can do to make this disease less pathogenic, and also understand what is happening. So very briefly, you all know this already, but I love to paint and I love to draw. So there's no better way for me to express myself about the disease than to draw the disease. So here's my drawing of the disease. So on the left side, the top are the green is the virus. Virus enters the lung. It grows in the lung. Inside the lung, there is a single cell, an epithelial cell. It grows inside the epithelial cell by infecting through the ACE receptor. All of these things you know, right? Then the virus enters macrophages, dendritic cells, macrophage, and then it induces this massive inflammation. And this inflammation results in affecting the heart, the gut, the brain, and so you see all these pathological effects of the virus. You know? So, and then finally you get multi-organ failure, which then results in death. So before we dive into the vaccine, let's start looking at what the immune response to the virus is. And there are three components of the immune system. One is the innate immune response. The second is the adaptive immune response. And the third is mucosal immunity. So I'll cover all the three aspects for understanding how this virus works. How many of you have thought, what is the immune system of a fly? Does a fly have an immune system? Of course, it does have an immune system, but it is not that evolved. It is not evolved like it is in humans. It has a very primitive and old immune system. That immune system is very simple. The fly has these receptors on the cell surface, which are called toll-like receptors or toll receptors. These receptors, they bind to the bacteria and to fungi that the fly can get infected with. And then these signal transmit biochemical signals into the cell, which then activate genes such as enzymes and toxins. And these enzymes and toxins kill the bacteria in the Drosophila, or this fly. And this is how the fly has the immune system. Interestingly, humans have the same toll-like receptor on their cell surface of macrophages and granulocytes. Same toll-like receptor, which is conserved from the fly. Same receptor. This is called the innate, very primitive immune system. It's the innate immune response. We also have toll-like receptors on the cell surface of these cells. They also transmit signals through biochemical mechanisms. They activate genes such as interferons, right? And these interferons secreted by the immune system will expand, and they will have multiple functions. So induction of interferon is a very important phenomenon in induction of an immune response to any virus, and especially this virus. So let's look at what this virus does. So I told you before that if there is a virus, it enters the cell, and the toll-like receptor I showed you in the previous slide is present on the cell surface. But there are also toll-like receptors in the intracellular. These are called intracellular toll-like receptors. So the virus binds to these intracellular toll-like receptors and transmit a signal, which then can activate the interferon gene in a normal situation. But this virus has at least 5 genes that it expresses that blocks interferon-induced signaling. It blocks it. So the NS4A, 4B, non-structural protein 15, M protein, the ORF3A, ORF6A, these proteins of the virus, they bind to and block the signaling mechanism downstream of the toll-like receptor and don't induce, and blocks the induction of interferon. Just think about this. Interferon is not allowed to be expressed by this virus in a very systematic way. And if you don't have interferon, you will not have immunity against this virus. So the virus has a capability of blocking interferon infection. There are other cells that are cells that secrete interferons, like natural killer cells and gamma delta T cells. What it turns out, and this is we have to still study this and prove it, people who live in countries or places where we are infected with a lot of different pathogens, like for example in India, right? We have a very high percentage of these natural killer and gamma delta cells. Just as an example, if you take gamma delta T cells in a clean population, like let's say in US or Europe, and if you take an average number of gamma delta T cells in that population, it is generally in the 1 to 2% of the cells will be gamma delta T cells. In India, if you take a population of people in India, we have 10% of our lymphocytes as gamma delta T cells. So if we have a larger number of gamma delta T cells in our population, we have an advantage against this virus because we have more cells that can protect us from this virus. And remember, I just showed you that the virus has a mechanism to block interferon induction. So this immunology is so beautiful, and it has to be studied in a very systematic way. And this is observation. This is just observation. मेरे पास कोई lab नहीं है. I'm not doing any experiments. I'm just reading 50 to 100 papers a week. And when I read all of this, I have to integrate all this information to make a story. I don't know if this story is true, but it is a hypothesis that can be tested. That higher number of interferon gamma secreting cells in patients, in people who are exposed to a lot of viruses, may be protected from this infection. It can be tested. And then finally, what you want to do in this situation is you want to look at a detailed analytical analysis of all the immune cells and find an immune signature that gives you a prognosis of the disease, which means if you have a particular kind of immune system that you have these CD4 cells, these CD8 number of CD8 cells, etc., you will be more likely to be protected than if you don't have this. So it's a beautiful way to study the pathogenesis of this disease. So this is the basic immunology that I was trying to tell you before I talk to you about vaccines. Remember, interferons are suppressed. We'll come to that a little bit later. So one background slide about we talked about the innate immune response. Now we'll talk about the adaptive immune response. The adaptive immune response is that is induced where you get activation of CD4 cells, B cells secrete antibodies, and cytotoxic T cells become killer cells. You need all kinds of cells to have an effective immune response against this virus, right? So the antibody response is called the humoral immunity, and the cell-mediated immunity is manifested by CD8 cytotoxic T cells. Humoral immunity means you develop antibodies which secrete it and they block the virus from entry. Cytotoxic T cells cannot bind to the native virus. They bind to virus that is being presented by infected cells, like for example, the lung epithelial cells. And then when they recognize the virus peptide, these cells will kill the infected cells. So this is called cell-mediated immunity. This becomes very important in developing a good vaccine. So remember, both arms of the immune system are required to eliminate the virus. The humoral immune response, which is the antibody response, as well as the cell-mediated immune response, primarily by cytotoxic T cells. So before we go further, we have to understand how is cell-mediated immunity generated and how is antibody response generated. So what happens is when a virus like COVID-2 enters the cell, an antigen-presenting cell, it is an intracellular antigen. And that is because it encodes its RNA, and this RNA is converted to protein. This protein is broken down into peptides, and these peptides are presented on the surface of MHC class I molecules, which then present antigen to CD8 T cells. These CD8 T cells, they differentiate and become cytotoxic lymphocytes, CTL, which manifest the cell-mediated immune response. And this is what a CD8 T cell looks like. This is a T cell which can kill a virally infected cell by that recognition molecule that I mentioned. These are just scanning electron micrographs of a T cell killing a target cell. On the other hand, extracellular antigen has to be taken up by antigen-presenting cell and is presented on a peptide to CD4 positive cells. And the CD4 positive cells, when they get activated, they help B cells make antibodies to the virus. So these are extracellular, and this results in manifesting of the humoral immune response. And the humoral immune response primarily is mediated by antibodies. And the antibody structure is like the Y structure, the heavy and light chain that you're all familiar with. And it has two binding domains. One is called the FAB, which is the antigen binding domain, and the FC portion, which is the stalk of the antibody. Yeah, we'll come to this and why I'm telling you this as we look at the vaccine. So now again, both arms of the immune system are required to eliminate the virus. As far as regulation of the immune response is concerned, it's a very interesting biology, right? So what happens is, on the one side, you have cell-mediated immunity, and the other side, you have humoral immunity. And I've just drawn it as a casual picture, but it is much more than a picture. What happens is, when you start an immune response, you start in the middle. And depending on the nature of the antigen, whether it's intracellular or extracellular antigens, whether you activate CD8 T cells or CD4 T cells more actively, you can either get a cell-mediated immune response or you can get a humoral immune response. So all the vaccines that we have got so far, for example, measles, mumps, rubella, hepatitis A, hepatitis B, all these vaccines that we get, right? They activate the humoral immune response. They activate CD4 cells, and CD4 cells help B cells make antibodies, and the antibodies make neutralizing antibodies, and that's how all of these vaccines work. In the history of vaccination, there is only, that I know of, there is only one vaccine that we have made where you have to activate a cytotoxic T cell, and that is the chickenpox virus, varicella. This is the only vaccine that we have made in the more than 100 vaccines that have been made in the world that is specifically activating T cells. We have never made a T cell vaccine ever, successful T cell vaccine in life. And what I will show you, COVID will require a T cell vaccine. And we have never made one before. This is why it's going to be extremely difficult to make a vaccine for this virus. And I'll come to why that is the case. Immunology, relative to other fields of biology like physics and mathematics and chemistry and even biochemistry and even molecular biology, is a very, very new field. It's only 20, 30 years old. We are studying immunology only in this depth only for the last 3-4 decades, not much. Highlighted by the fact that Max Cooper and Jacques Miller received the Albert Lasker Award for discovering T and B cells last year. Lasker Award is the equivalent of the Nobel Prize in the United States. It's a United States Nobel Prize. It's called the Lasker Award. And they got this award for discovering T and B cells last year. So you can imagine how new the field of immunology is. And we need to learn immunology to make a vaccine against this virus. So we have so many challenges, right? It's a beautiful time to be in the field of science and study biology. You're in a fantastic place, right? Study a lot of immunology. The last thing I'm going to talk about in immunology for this virus is the mucosal immunity. What is mucosal immunity? It is the immunity from the nose, from the knees. The virus comes through the nose and mouth, right? That's how the virus comes. So you have to study what the immune response in the mouth is, in the lung is, the gut, and the genital tract. All of these exposed surfaces in our body are the mucosal immune response. And I will touch upon this in the vaccine. I'm going to quickly skip over the virus because I think you all know about the virus and you've already had a presentation on this. The virus contains many proteins. All of you have heard about the spike protein, I'm sure. This is what the spike protein looks like on the cell surface of the virus. It has two domains. It has a stalk and it has a surface. Now what happens is the stalk protein, the spike protein has two components, S1 and S2, and there's a proteolytic cleavage site in the middle. Proteolytic cleavage means it cuts the portion, and that's about here. You know, the red arrow, it cuts the enzyme cuts the spike protein at that position. In a cartoon, this is what it looks like. There are 3 steps in the entry. The virus has to first bind to the ACE2 protein. We all know that. Then the spike needs to be cleaved from S1, S2, and then the fusion takes place. And the cartoon that I will show you does that. So it first has to be cleaved and then enter. Let me show you that again. The virus has to bind to ACE2 receptor. S1, S2 have to be cleaved. If this cleavage doesn't occur, the virus cannot enter. And then it enters. So it's a very complicated procedure to enter the cell. It is not a simple entry. So the vaccine should induce an immune response not only to the red portion of the spike, but also to the purple portion of the spike. This is not an easy task to do. The good news is that a lot of research has suggested that when you have antibodies against that ACE2 binding section of spike, you can actually prevent virus from entry in an in vitro setting, right? So what do we know about the virus immunology so far? All patients exposed to the virus develop antibodies. Antibodies are directed against spike protein in the patients. And antibodies to the spike protein can block virus entry in laboratory-based experiments. We just showed you that. What is in progress? A lot of things are in progress. What is the level of antibody that is protective? We don't know that yet. Whether both humoral and cell-mediated immunity are required for protection, we don't know that yet. I told you about the FC and FAB portion of antibody. So neutralizing antibody is an antibody that binds to the virus through the FAB portion and then blocks the virus from entering the entry into the cell by blocking the interaction with ACE2 receptor. That is a simple thing to understand, right? Non-neutralizing antibodies are antibodies that bind to a non-ACE2 binding portion, non-receptor binding portion of the virus. So what do these antibodies do? And just focus on this side. When an antibody binds to the virus, the FC portion binds to what is called the FC receptor on other cell types, and it will enable the virus to infect ACE2 negative cells. The virus is infecting ACE2 cells. If you develop an antibody response by the vaccine antibody will bind here and then enable the virus to infect non-ACE2 positive cells. So there is a possibility that the vaccine will make the virus more infectious. There's a theoretical possibility. These are called antibody enhancing antibodies, or virus and antibody enhancing antibodies. Okay, so this is a very careful thing that has to be seen. A vaccine may make the disease worse. The theoretical possibility. We know the course of infection now, that there is an asymptomatic phase where the virus grows, and this is where a lot of transmission is happening. There's virus load, and there's symptoms. You finally get IgM and IgG antibodies, and you get T cell responses. And the goal of the vaccine is to develop antibody and T cell responses. So now let's switch gears a little bit. Let me pause for a second because I've presented a lot. And so what I've covered so far is I showed you a little bit about the innate immune response. I talked to you a little bit about the adaptive immune response, the role of cell-mediated and humoral immunity. I told you that the mucosal immune response is important. I also told you that the spike protein develops a neutralizing antibody, which can be very good for developing a vaccine. So that's where we are right now. And let's look at how the vaccine is developed. So what is the question that we are asking for the vaccine? What is the nature of the protective immune response? Is it humoral immune response or cell-mediated immune response? What part of the virus can induce the immune response? What kind of vaccine do we need? How frequently do we have to give the vaccine? How much dose do we have to give the vaccine? We have to study all of this. And how much vaccine needs to be made? So in the field of drug development, this requirement of what you need to make the product is called a target product profile. It means it's just a form that you fill to say, okay, what do I need? What do I want? Main kya bana raha hu? Design time mein, what am I making? Right? What are the features of the vaccine? So what is the goal of the vaccine? The goal of the vaccine is to prevent infection. But it is also to stop replication. The indication will be most likely first patients who will get it, first people who will get it are healthcare workers like doctors and nurses and all the people in the hospital. They should get the vaccine. Susceptible individuals like people who have comorbid conditions like cardiovascular disease or diabetes, they should get the vaccine. And then the healthy contacts of people who are in contact, who have family members who have the virus, they should get the vaccine. So that number, approximately, anybody's guess, it could be anywhere between 10 to 100 million people. It could be more. Who knows? It's a very large number. We know that. What is the measure of efficacy of the vaccine? That it should elicit a neutralizing antibody, and it also induce a cytotoxic T cell response, CD8 T cell response. We have to determine what the dose is. This is the approximate dose. It should be durable at least for a year. And then the vaccine should be stable at least for 6 months. You have to distribute the vaccine all over the world. It should have stability. It should not degrade. In the development of vaccine, which usually takes 10 to 20 years, we as in humanity would like the vaccine to be generated in the next 6 months, but realistically, it is probably going to be about 2 years before a lot of vaccine is made. The process of developing a vaccine involves these steps. Antigen identification, and we know it is spike. We think it is spike. We don't know, but we think it is spike. The spike is expressed in different ways: RNA, DNA, protein, viral vector, inactivated, live attenuated. You have to develop a process to manufacture the process. Then you have to do animal studies, clinical trials, regulatory approvals, and then of course you have issues with scale up and distribution and all that. So this is the life cycle of vaccine development. What we've done in 10 years, what requires 10 to 20 years, we've done in months. There are many vaccines where the clinical trial has been initiated. I will talk about the Oxford chimp adenovirus vaccine just as an example. I'm not saying that this is the best vaccine or anything yet. We'll look at the results as an example of what it takes to develop a vaccine. So I'm presenting that in that regard from the published data that is out there. So the ChAdOx vaccine is a chimp adenovirus candidate vaccine, which is an adenovirus, chimp adenovirus, that is expressing the spike protein of COVID. So what is taken, the viral proteins of adenovirus are removed and replaced with the spike protein of COVID. The Oxford group has injected such a vaccine, not for COVID, but other diseases like Ebola and others, in about 300 individuals. So they know it is safe. And it induces both humoral and cell-mediated immune responses. I showed you this slide previously. And in terms of a vaccine, it does everything that the virus does, except that it is not infectious. So the vaccine comes in, induces activation of CD4 cells, and these CD4 cells will help make antibodies to the spike. The vaccine will also activate cytotoxic T cells because it can activate cells through the intracellular pathway and activate CTL, which will be important. So now on 13th of May, the ChAdOx group published a paper showing the results of the vaccinated studies in the monkeys. 6 monkeys were vaccinated with the COVID vaccine, and 3 were given a control vector which expresses green fluorescent protein, which is not a anti-COVID vaccine. The injection was the intramuscular injection. They measured immune responses, and after 28 days, the animals were challenged with a live COVID-2 through the intranasal route. So that is the study. So this is the result. The vaccinated monkeys develop immune responses as expected. All the red dots are the vaccinated monkeys. The blue dots are the control monkeys. And we are measuring antibodies to the neutralizing antibodies, virus neutralizing titer. And you can see that the vaccinated monkeys are developing neutralizing antibodies. Spot forming cell unit is a measure of cytotoxic T cells, CD8 cytotoxic T cells. And these, you can see that the vaccine is also inducing cytotoxic T cells in some, but not all the monkeys. Yeah? More importantly, these CD4 positive cells, this is cell-mediated immunity, are secreting interferon gamma. Remember I told you this virus has a very specific mechanism of blocking interferon. So this vaccine is inducing interferons in these monkeys. So it's a good sign. So before we look at the results of the challenge study, let's look at the mucosal immune response. So what is the mucosal immune response in this virus? It is the upper respiratory tract, which is measured by taking a nose swab, right? And that is the nasal cavity, the larynx, ye line ke upar, all the things are considered upper respiratory tract. And the lung and the deep lung are considered lower respiratory tract, for which the sample that is taken is called the bronchoalveolar lavage, which means you put a tube through the mouth and you collect a sample from here. So that is a bronchoalveolar lavage. And now in the next slide, busy slide, but I'll show it in your box. Let's focus on the middle box. They're looking at the bronchoalveolar lavage fluid from monkeys who are vaccinated and not vaccinated. And they are looking for the amount of virus that they can measure after the intranasal challenge. And in general, you can see that the red dots, which are the vaccinated monkeys, are lower level of virus compared to the blue dots. That means the vaccine is protecting against the lower infection. Okay. Look at what happens to the clinical score, which is measured by the clinical symptoms of the virus. You can see that the vaccinated monkeys are getting infected. It is not like the vaccine protected against infection. The vaccinated monkeys are in fact getting infected. The thing that happens in the vaccine is that the vaccinated monkeys seem to clear the virus a little bit faster than the non-vaccinated monkeys. The vaccine is not preventing infection, even though there is neutralizing antibodies. And that is because you can see that in the nasal swab, upper respiratory tract, there's no difference between red and blue dots. So the vaccine doesn't protect against upper respiratory tract infection. At least this small study shows that. Whether this will play out in the clinical trial, we don't know. I hope that in the clinical trial, it shows much better, but this is what the current data shows, right? The vaccination prevents against pneumonia, for sure, for lower respiratory tract. So it's a good vaccine for preventing pneumonia so far. This is just a pathology of the monkeys, where you look at the tissue and you can see infiltrating lymphocytes in the control monkeys, and you get a nice clean tissue distribution in the vaccinated monkeys. So what is the summary of the vaccination so far? Is that the vaccinated monkeys induce both cell-mediated and humoral immunity. It does not induce sterilizing immunity, because the vaccinated monkeys do get infected with the virus. But the vaccinated monkeys are able to clear the virus a little faster than the control monkeys. It prevents lower respiratory tract infection, but may be less effective in the upper respiratory tract. And then it prevents pneumonia. There are many other vaccines that are doing that. The results are out for. I just showed you the example of Chadox because as an example, but there are many more data from many other vaccines, and a lot of data is being published. But generally, all the vaccines so far are showing similar kind of observations. Sorry. The process of manufacturing a vaccine is a whole different ballgame. You need mechanical engineers and biochemical engineers, and you have this massive facility. So the scale of developing the kind of talent you need for making the vaccine is very different from the talent that you need for designing and testing the vaccine. You need biologists and molecular biologists and immunologists and all that in the first field. Here you need engineers and all that kind of, those kind of stuff. So the process involves is huge facilities, and this is how a vaccine is made in large scale. And Serum Institute is one of the premier institutes in India that is developing, but there are other places like BioE is also developing. But there are many other companies who are doing support of the vaccine, which is very critical. Making the vials, making the rubber stoppers, making packaging equipment. So many jobs are there in the vaccine manufacturing development, not just making the vaccine, right? So as you're looking for jobs after you're finishing your college, there are hundreds of jobs in this field, not just science. Okay, one interesting component now when we do the clinical trial, which is starting now, is the clinical goal of the vaccine is to develop a protective immune response. The immunological goal is to develop neutralizing antibodies and cytotoxic T cells in healthy individuals, and these are the clinical trials that are started. So how do you test for whether the vaccine works or not? Volunteers will be blinded. They won't know whether they have received the vaccine, or in the Chadox vaccine clinical trial, they get the control arm is the meningococcal vaccine. And after it's a blinded study. So after a predefined number of people are infected, the study will be unblinded. Just one note before we go to the results, what are the potential results of the clinical trial? It is interesting that the bioethics groups have actually weighed in on the fact that in a human clinical trial, just like in monkeys, it may actually be possible to really infect some people with the virus. Although it sounds very unethical to actually take the virus and infect people to test whether the vaccine works or not. But that may be a very fast way to determine whether the vaccine works. And the ethics committee has weighed in on this, and there's a very nice paper written on this topic. There are advantages. The process is like this. The preliminary testing is done to ensure that the vaccine is safe, and relatively healthy people will be given the virus. They will get either the vaccine or the placebo. You wait for some time, and then you expose them to the virus. This sounds very dangerous, right? There are risks, but it has been done before. Influenza has been tested like this, typhoid, cholera, malaria. It has been done in humans. People have been given, it's not like this is the first time something like this is done. It's been done before. There are risks, but the risk can be reduced by giving young people and who are relatively young people this virus. And then you can also select people who are already in a very endemic area, unko वैसे भी होने वाला ही है. Of course, you need informed consent and the whole process. You need that, but there is a very interesting phenomenon that this is possible to do. We'll see. But anyway, in the normal course of the vaccine clinical trial, you have to do phase 1, phase 2, phase 3 studies, write a huge report. The evaluation will be done by the regulatory agency, and then you will get approval. So now let's say the clinical trial is done, and what can the possible results be? So I told you that the design of the clinical trial will have a group of patients who are vaccinated and a group of patients who are not vaccinated. And you have to compare. It's a blinded study. Now, after the blinding is open, after 100 people are infected, you want to find out where those 100 people fell. If those 100 people are all the infected people were in the meningococcal vaccine arm, the efficacy of vaccine is 100%. If 20 of the COVID vaccine patients got COVID infection, that means the vaccine is 80% successful. If 70% of the subjects only are in the meningococcal arm, then your efficacy is average. If there is no difference, if 50 patients of the 100 people fall in the COVID and 50 fall in the meningococcal, then the vaccine doesn't work, that means it's failed. But there's one more possibility. More people could be infected, more of them could be in the vaccinated arm than the control arm. Then the vaccine will be dangerous. And why am I showing you this? Is because for dengue and for some other viruses, this phenomenon has been demonstrated. That when you vaccinate people, you actually make them more susceptible than if they were not vaccinated. There's a theoretical possibility. Although the early animal trials have suggested that this is not the case, there's a theoretical possibility that this can happen. So we'll be very careful with the vaccine. Not easy to make a vaccine. Finally, a successful vaccine should be efficacious. It should induce a mucosal immune response. We studied that, right? I showed you that. It should have no or minimum side effects. And it should be economical. It should have social impact and, of course, political impact. Finally, last slide. What will the vaccine do? It will induce herd immunity. And what is herd immunity? In a population, you will have this group of people. You will have uninfected people shown here in green, infected people shown in red, and infected people who are hospitalized who will be sequestered. Black dots will be the people who are previously infected and therefore they will have neutralizing antibodies, and they will have, and then you will have the vaccinated group who are in blue. So just look at this red dot in this middle. This red dot infected person will infect this green dot uninfected person. There's no choice. That person will definitely be infected. We know that. If you get exposed to this virus, you will get infected. That is why we have social distancing, right? So once this red dot infects this green dot, these two will become infected. Now what will happen after that? They cannot infect anyone around them in this picture because either they are previously infected or vaccinated. So the virus will die in this population. This process is called herd immunity. And we don't know today whether 30% of the population or 80% of the population needs to be vaccinated or infected before we get herd immunity. So a lot of modeling studies are in progress to determine what is that number of people that need to get infected before you get herd immunity. So this is a very interesting mathematical problem that we are trying to solve. And so I'll end here by saying that in today's time, there's an explosion of science. I showed you a little bit about basic immunology of the virus, the components of the virus, the immune response to the virus, the rationale for the design, and the herd immunity. So I'll stop here and thank you very much for an opportunity to present. I will unshare my slides and take questions. Thank you so much.